Oh, thank you, Bob. And I'd like to thank Tony, excuse me, and Beth as well for organizing this meeting and giving me the chance to tell you about some of our research. So my lab has long been interested in the general question of how it is that the brain can selectively access the information in cerebral cortex that is best suited to handle the task at hand. And for most of my career, that work's gone on in the context of studying attention in trained behaving rhesus monkeys, generally visual spatial attention. But about 10 years ago, when we saw how powerful the genetic and optogenetic tools were in mice, we realized that there were a lot of experiments that we could do and really wanted to do that would be based on optogenetics. And at that point, we decided that we were either going to have to devote a large chunk of the lab's effort to refining and developing some of these genetic tools for monkeys, or else try to refine and extend the behavioral tasks that could be done in mice so that we could pair them with the sort of monkey experiments that we were doing. For us, it wasn't a long discussion. We have a lot more experience in training animals to perform tasks than we did in molecular or genetic biology. And so we took the approach of trying to bring mice as behavioral prep that would allow us to sort of dovetail with the work that we're doing in primates. We've had some success there, but run into some limitations as well. And so I thought for this meeting, it might be a good idea for me to just run through what we've been doing with the mice, what we've achieved in terms of their behavioral performance, what sort of barriers that we've run into. So I will highlight both their strengths and weaknesses. And then turn to some of the more recent work we've been doing in monkeys, just to sort of give you an example of the types of information we're trying to extract from these behavioral studies to give some clarity to why we're running into some problems with the mice. And I would say developing the mouse prep was largely effort by Mark Heisted, who was a postdoc in my lab at the time. He now has his own lab at NIMH. When we started, we knew we needed head-fixed animals trained to perform tasks similar to the ones that the monkeys were doing. At the time, and even now, I think the most popular approach is to put a head-fixed mouse on a treadmill or on a rotating ball. We thought about the implications of putting a head-fixed monkey on a treadmill or a rotating ball, and we just decided that that was a source of uncontrolled variance we wanted nothing to do with. So our mice aren't floating in the air. The approach we've taken is a little bit like the mouse prep. So this is a head-fixed mouse. He's got his little hand on the lever, which he can release at the appropriate time. He's viewing a screen. Over here, you may see gratings appear occasionally. His job is just to release the lever when he sees one appear. And as with the monkeys, we use tones to give the mice some feedback on their performance and also so we can hear what they're doing. Mice can get pretty good at this sort of task. This animal was trained for about 2 months, and good animals, after about 2 months of training, are pretty reliable psychophysical observers and will give pretty reliable data. But I was a little surprised. That's pretty much how much time I would allocate for taking a naive animal, a monkey, and getting them up to becoming a good psychophysical reporter on a simple task like this. So good for the mice on this part. Once they are well trained, their performance is just fine. So these are just sort of typical data from a good mouse, just responding to a high contrast grading. There's a fairly low level of false alarms. There's some misses in here. Typical lapses are 5 or 10% for something like that, but a nice sharp reaction time distribution. And with that sort of performance, it's easy to sort of adjust the contrast of the stimulus that you're presenting and get contrast response functions from the animal to just sort of measure their contrast thresholds for detecting the onset of stimuli on the screen. And their performance is pretty good and pretty consistent day to day. We lose about half the mice we train. They just drop out along the way for not performing well, but a good fraction of the animals become pretty proficient. These are just 6 sessions from 3 animals and just sort of show the level of replicability that we can get. Pretty good thresholds. 1 bad day in here. Mice sometimes go through bad days or bad weeks, but in general, it's pretty obvious when they're working well. And when they do work well, you can get 300, 500, maybe 700 trials a day, which is enough to get a good psychometric function, or 2. And 2 is important because it allows you to do a perturbation and compare the effects within a single day. When animals are working this well, we can adjust parameters like the spatial frequency and measure the spatial frequency functions for the mice. So here's a contrast sensitivity function for 3 different mice as a function of the spatial frequency of the stimuli presented. And you can see, again, quite good consistency between the animals. With performance falling off at about 1 cycle per degree. 1 cycle per degree is pretty low acuity, and here's a good point to just compare this to human and rhesus monkey performance. So, the testing conditions were different in all of these, so they're probably not directly comparable, but the roll-off here really is true. So human monkey performance is rolling off at about 10 cycles per degree. Mouse is somewhere between 25 and 50 times worse. And so they really can't see this stuff. And that's a simple function of the number of retinal ganglion cells they have. I don't know if the peak is for real here or whether that's testing conditions, but they have far fewer photoreceptors, so you could expect that that could also be a real function. But the thing I want to point out is just this gap here. Mice don't see that well. What they do see, they're fine, so you just need to stay out of that range. But I think it's an important thing to point out because it means you really have to stay in their sensory range. And my impression after having worked with the mice is that there is a comparable cognitive gap for the mice. Just as they simply have no representations of high spatial frequencies, I think there's a whole range of cognitive tasks for which they have no representations and just simply are unable to perform. And attention is key in my mind here. We'll come back to that a little bit further. But they're champions with genetic tools. So, as someone with minimal experience with genetics and molecular biology, the idea that you can buy mice from a catalog and buy virus from a catalog and inject the virus and get it to express and shine light and get effects, I find just really amazing. And it works like that, off the shelf, for even biologists like me in my lab. So, for example, I'll run through just some of the things that we've been doing lately. One of the things you can do is buy mice that selectively express Cre in one class of inhibitory interneuron, parvalbumin cells, somatostatin cells, vasointestinal peptides expressing cells. You can then inject AAV, virus, that will conditionally express channel rhodopsins in cells that are expressing Cre. And so you can load up these specific classes of interneurons with channel rhodopsin, and then when the visual stimulus appears on the screen, on a randomly selected subset of the trials, just shine a light, typically a few milliwatts of light onto the cortex. These are injections we've made in V1, so we're disrupting the representation in V1, and measure the consequences on the animal's behavior for doing those sort of manipulations. You probably aren't surprised that turning on inhibitory neurons shifts the contrast function for the mice. Their behavior is worse, their thresholds are higher, both for the PV cells and the somatostatin cells, in every session that we tried, in every mouse we ever tried. That's probably what you should expect. Those cells reduce the number of spikes in the pyramidal neurons. You might not expect that the VIP cells would have the opposite effect, but they do. But in fact, that's expected. A major effect from the VIP cells is to suppress the somatostatin cells, increase the number of spikes in the pyramidal cells, and so you get the opposite effect, basically a disinhibition. But it's very straightforward to measure fairly robust changes. These are individual days and individual mice, and you can see that it's pretty reliable data that you can get from these animals using this sort of approach. The data are high enough quality you can actually address specific questions about what are these types of effects. For example, in single-cell physiology, patch clamping in vivo, looking at how these inhibitory interneurons affect responses in cortex, is a bit of a debate about whether the effect is additive or multiplicative in terms of the number of spikes that are produced. You can see here in terms of the behavior, it looks like a pretty straightforward rightward or leftward shift on this log axis, which would be consistent with a multiplicative effect on the perception of contrast. That is, these inhibitory neurons seem to have a gain control effect. They are sort of multiplicatively changing the effective contrast, whether it's a high contrast or a low contrast. In fact, when we test multiplicative models versus additive models, where changing the inhibitory neurons would add or remove a fixed amount of effective contrast, there's a clear win for the gain models. It seems quite clear that what these inhibitory neurons are doing perceptually for the animals is changing the effective gain of the contrast that's being presented to them. These are all experiments that we've done where we've continued the animal working with a visual stimulus and perturbed his ability, her ability to detect that stimulus. You can take the stimulus away entirely. So it's long been known that electrical stimulation of human or animal brains in visual cortex will produce a phosphene. Optogenetic stimulation of V1 in mice presumably produces a phosphene as well. And so if we take an animal that's well trained on performing the visual task, showing here the performance in terms of percent correct and reaction time, and then just remove that stimulus entirely and have them respond to the occurrence of optogenetic stimulation, where we vary the power from very low to very high, we can get a psychometric function of detecting this optogenetic stimulation of V1 that has similar sort of sensitivity. The slope is comparable, and it's occurring at relatively low power, although the power is really a function of how many cells are expressing, how many virus did you put in, so that's not really a veridical sort of measure. But the fact is that with fairly moderate intensities of light, we can get strong responses behaviorally. We also get premium on the reaction time. When you do the optogenetic stimulation, reaction times are faster for similar levels of performance, presumably because we're bypassing that sluggish retina and activating things directly in cortex without any of the latency of the retinal processing or the earlier steps going on. Well, with this sort of approach, we can actually make some good progress characterizing the readout mechanisms of cortex, the spatial temporal integration. And just one example is the temporal integration. So we've looked at whether it matters to a mouse if we deliver all of the power in a very brief pulse, or extend that, or extend that. And here we were just delivering power either in 100 milliseconds, 10 milliseconds, or 1 millisecond, and looking at how much power we needed to deliver to get the animal to detect it. And you can see the animal could work at any of those powers, but needed substantially more power for the shorter durations. But you can see the shifts are about an order of magnitude corresponding to the durations that we presented. And in fact, if you integrate the power delivered for all of these functions, they pretty much superimpose. That is, there's a trade-off one-for-one between the power delivered and the duration of that stimulus delivery. In fact, when we did the physiology in these animals, the number of spikes also was more or less pretty constant whether we delivered the power in a short interval or not. The implication is that the animal's performance more or less depends on how many extra spikes we put into the cortex, at least over periods from a few milliseconds up to 100 milliseconds. That is, it's like the animal's a perfect integrator of spikes in terms of being able to detect what's going on in that region in cortex. We didn't go longer because the reaction times were short enough that we couldn't actually explore further. But these genetic tools allow us to look at these sorts of questions about temporal integration. And there's equivalent questions that we'll be looking at in terms of spatial integration as well. Does it matter whether it's a large area that's stimulated or a smaller area? And we're currently part of a consortium that's trying to develop methods for two-photon stimulation, which will allow us to pick and choose individual cells in the field of view and ask, how they're distributed or in different layers. And, I think there's no question that the mouse will give us a lot of power in those directions. But at the same time, I've been disappointed in other ways in terms of what we've been able to do to the mice. We can bend them to our will in many ways, but coming back now to this sort of gap between the sensory performance of mice and primates, we've also really had some trouble trying to get them to do the sorts of tests that we want them to do for attention studies. And going into this, I thought visual spatial attention was going to be pretty manageable for mice. It seems like a basic sort of function. There's even physiology in insects suggesting that they could do it. But when we got these mice in the rig and tried to train them to pay attention to one side more than the other side by adjusting reward scheduling, we really had a tough time with that. We got no real progress until we just sat the mice down, 90% of the rewards on the right, 10% on the left, for 2 weeks. And by the end of that time, we were starting to see something we could measure in individual days, that they actually had a little more sensitivity on that side. And then we flipped it and went on for another 2 weeks. And finally, at the end of those 2 weeks, we were getting an indication that the animal had more sensitivity on the side with the higher reward schedule. And it sort of shook me a little bit, because my feeling was, that's not what we're studying in monkeys. And, it's not even clear to me that it's the same mechanism. And that if we went down that road, I wasn't at all confident we'd be looking at the sort of attentional mechanism that we really wanted to study. And I think, we're not the best in the world. And in fact, I know other labs have had more success with attention. Lindsay Glickfeld at Duke is doing good work with attention. Rich Krauslis at NEI is doing good work, but my feeling is it's still a very different animal than the sort of cognitive function that we want to study. And part of what drives me away is the questions we're now looking at in monkey are so far beyond just more left, more right, that I just feel like they really aren't going to be able to help us very much. So what I'd like to do is actually just show you the types of experiments that we're doing in monkeys to sort of help drive home why I've gotten to a position of feeling really lacking confidence that the mice will ever provide a useful model for us in terms of studying the circuitry of the phenomena that we've been studying in rhesus monkeys. And I would say where we are now with our studies of attention, visual spatial attention, is that we're really trying to understand it as a multifaceted phenomenon. And our model here actually is memory, and how what used to be one thing, memory, is now subdivided into very distinct, clear, obviously different mechanisms, which have been really mapped onto different neurobiological studies. And it's just not good enough to talk about memory in a general way anymore. You really have to talk about what type of memory you're talking about. And I'll say attention is still a little bit in the dark ages that way. We talk about attention, and I think I will show you some evidence that attention is multifaceted, that there are probably very distinct structures, and I think Rich Krauslis was a leading force in sort of emphasizing this, and that what we really need to do now is try and understand what are those different components that make up the attention mechanism that we've all been studying. And I think it's kind of critical, right? We now have, I think 8% of children between 2 and 17 in the United States are diagnosed right now with attention deficit hyperactivity disorder, and 2/3 of them are medicated for it. And I can't tell you what attention is. We study it a lot, and we're still always just using operational definitions. And so I think it's really an important step forward now to try and get at more clear-cut definitions. And so what I want to do is just take you through some work we've been doing, trying to distinguish attentional changes in sensitivity into components of selectivity and effort, which is, I think, the smallest sort of step that you can make. Just to be clear, the types of attention we're talking about here is visual spatial attentions, and this is sort of typical of the types we show our monkeys. If you want to play along, you just fixate on the white cross. The only thing that's happening here is that at a random time, the stimulus on the right will change its orientation by 90 degrees, and that shouldn't challenge you too much. It depends on whether you're closer or further away, but this is a pretty easy task. But you should get the sense that you have an ability to sort of cast your attention to one side or the other. In fact, with this sort of orientation change, you could cast your attention to the left and you'd probably still notice those orientation changes on the right. But I can change that. I can make it more difficult by giving you a smaller orientation change. So here, these orientation changes are about 15 degrees. And odds are you won't see them very easily unless you do have some more attention directed towards the right half of the screen. And with this sort of approach, it's relatively straightforward to train rhesus monkeys to do this task. They'll hold their gaze within a half degree of that spot. They will follow your instructions and actually show substantial changes in sensitivity on one side or the other as they do these sorts of tasks. We've looked at a number of different phenomena. The simplest form that attentional effects take in visual cortex is that if you have a single stimulus in the receptive field and the animal shifts its attention towards that stimulus, you'll generally see an increase in the responsivity of cells whose receptive fields are in the area the animal is attending. So here, for example, the average cell in V4 increased its response to all orientations by about 30% or so. What we've been doing lately is trying to get a little more subtle on what is changing when the animals shift their attention. And to just walk you through the sorts of approaches that we're using, I want to introduce you to D-prime space, a sensitivity performance space, which compares the performance on the left side stimulus with performance on the right side stimulus. And as I just showed you in those examples, you should expect that when the animal's attending to one stimulus, performance will be high there and low on the other side. And when he shifts his attention, that will invert. And that's exactly the sort of manipulation that's plotted here. we can generally run the animal up to a D prime of 2 or 3 on one side when performance is quite low on the other side. When the animal's attending to the right, if we ask him to attend to the left, we can see this shift in the other direction. I will say you have to work to get this. For many, many years, we didn't measure these. We didn't worry about it. We just looked at how performance was shifting on the one side. And I'll tell you, monkeys just want to have fun. They don't do what you want to do. And if you take a typical monkey, you'll often see that when you ask them to shift from left to right attention, they'll just keep attending a lot on one side and then add some more on the other and take it away. They don't do this sort of stuff automatically. So you actually have to monitor attention if you want to see what the animal is actually doing. Now, we wanted to look at effort. Attention has a selective component. Are you attending the left or the right? But you can attend a little or a lot. So when I gave you 90 degrees, you didn't have to pay much attention. But if you wanted to catch 5 or 10 degrees, you'd have to pay more attention. And you can crank that dial up or down. And that's sort of what we call attentional effort. It's been looked at a little bit in experiments. We've done some looking at effort, but the usual model is sort of what I gave you, just the task is easier, the task is harder. And what that does is it tends to move you further out along this diagonal. But this is a two-dimensional space, and there's actually orthogonal axes here, and it's entirely possible to take performance and move it in this direction instead. So here's a situation where there isn't much selectivity. Performance is equal on the left to what it is on the right. But you can go from relatively poor attention to both sides to relatively high attention to both sides. And in fact, this dimension really hasn't been studied much at all in neurophysiology experiments. And so we didn't know whether neurons in cortex are modulated by that effort or not. And so we wanted to examine that question. And so these are experiments that were spearheaded by Supriyo Ghosh, a postdoc in my lab who's actually here at the Colloquium today. And what he did was train animals to do a task that allowed him to independently control selectivity and effort in these animals. And the task was simple. The animal fixated, a pair of sample stimuli came on, they disappeared, and then randomly one stimulus or the one side or the other side would have a probe stimulus that either did or did not match the orientation on that side in the sample. If the orientation changed, that was a target. The animal had to make a saccade. If it didn't change, you had to wait until the next stimulus, which always changed. But by varying the amount of reward associated with each side and the probability that there would be a probe stimulus on each side, you could actually, with some effort, move the animal's attention around through high or low states. The simplest thing is just to run this task where the probe is equally likely on either side, equally difficult on either side, and then just vary the reward. If the reward is small, the animals won't work very hard. Their performance, measured in D prime, will be relatively low. If you increase the reward size, the D prime will improve. Supriyo found that you could do it either with reward or task difficulty. If you made the orientation changes much harder, the animals would have to work harder, and they did. And you could actually measure that their performance was better by inserting probe stimuli in the easy condition and the more difficult condition, showing that they were doing much better with probes in the more difficult condition. But the upshot was, in whatever method that this was manipulated by behaviorally, cells in V4 showed that there was an increase in the level of activity that's going on. So that they respond more strongly when the animal is putting more effort into the task, regardless of whether there's more selectivity or not. Now, you might think that's a relatively trivial result, because moving from here to here is basically saying we're taking the receptive field side and we're going from low performance to high performance. We're doing it on a diagonal, but why wouldn't you expect to see stronger responses from the cells if the animal's doing better in the receptive field location? So that just asks that we do this other comparison here. Is it really true that the activity of the cells is directly determined by the sensitivity in the receptive field location? And so Supriyo took the next step, which was simply to march the animals through this space, actually doing the equivalent of, I think, 2 laps on most days, taking the monkey through all of these different conditions to measure the responses of cells in V4 while the animal was doing this task. And these are actually representing behavioral data. The points are the averages. These somewhat too faint gray lines here actually represent the movements that the animal made in the course of a day, where behavior was controlled by reward or where behavior was controlled by difficulty. we're going from a D prime of 1 to almost 3 in these cases. This is really substantial changes in behavioral performance, something you would never get from a mouse, let alone the movement through the space. And so the question here was, what was the neurophysiology like? And the answer was interesting and not simple. So you might expect that the inside receptor field D prime would be closely related to the neurophysiology. The blue and green would be high, the yellow and red would be low. Ballpark true, but you can see the green and yellow are not the same as the blue and the red. For example, going from the green to the blue reduced the level of firing in V4, even though the animal's performance was exactly the same in those conditions. So the performance of the animal was not changing, but the rate of firing for the cells in V4 representing that part of the visual field was there. So clearly, V4 cells depend not only on the selectivity that's going on, but also the sensitivities. And there's interactions that are occurring here that we still need to sort out. So attention, which we previously had just described as attention to one location, really does depend on how selective that attention is and how intensive that attention is. In the last part of the talk, I'd just like to mention some optogenetic experiments that Suprio has begun in monkeys. And we never started with optogenetics in monkeys because it just has not been as well developed as in mice. And there are good reasons why that's the case. There are, for example, transgenic lines in mice because they are so cheap and breed so readily and their genetics are so easily manipulated that it's possible to buy animals that express Cree in different cell classes. But beyond that, there are probably more than 100 times as many mouse labs as monkey labs using viruses, maybe many, many more. And the fact is, all those mouse labs are refining things and working things out and figuring out the right serotypes and tropisms, and basically they've sort of fine-tuned all of those viruses. In the monkeys, we'll never get there because we'll never have as many labs working on that or as much histology to validate the methods, but things are improving and there's every reason to believe that they will continue to improve in the long run. But Suprio wanted to take the step and see where he could get the optogenetic approach working in monkeys. It seemed like a rather choice thing because this effort component that we're talking about in attention seems like it might be related to activity in the locus ceruleus, which has long been associated with arousal and attention and other sorts of functions of that type. And genetically, that's one of the more approachable structures because it's got neurogenergic cells that innervate most of the nervous system. And the neurogenergic component just means that it's easier to target that structure. Not easy, but easier. And so Suprio injected one virus, which basically caused cells with DBH, the protein that converts dopamine to noradrenaline, to express Cree. And that basically produced expression of Cree throughout the locus ceruleus in one side of the brainstem. He then injected another virus, which caused Cree expressing cells to express channel rhodopsin. And then tested the monkeys, performing the type of task that I just described, with and without stimulation of the locus ceruleus on one side of the brain. And there was a definite and positive effect of that stimulation. If there was stimulation when the animal was attending to and working with the stimulus on the ipsilateral side, there was no real effect. But when the animal was working with the stimulus that was contralateral to the locus ceruleus stimulation, there was an improvement in performance. The curve shift to the left, thresholds were slightly better for detecting an orientation change in that condition. If we plot the net result in D prime space, for the contralateral and ipsilateral location, you can see what's happened here is basically D primes moved up for the one side, whether it was in a lower attention or lower effort or higher effort condition. Got no appreciable shift for the ipsilateral location. So this was an interesting sort of effect. It was cranking up the performance behaviorally on one side of the brain, one side of the visual field. The fact is, we don't know if the locus ceruleus ever works in this mode where it has differential activity on one side relative to the other. I had always assumed that it was sort of a a tightly coupled structure. We're perturbing it, but you know, in principle, there's no reason we couldn't perturb both sides or suppress both sides. But I'm very pleased with this result because it's the first optogenetic success that we've had. Optogenetics in monkeys has been a little troublesome because, for reasons that aren't really understood, it's never been particularly efficacious. So whereas 25 microamps of electrical stimulation will get you a saccade or a perceptual report or other types of measures on individual trials on a completely reliable basis, to my knowledge, and I'm not completely up to date here, I don't believe any optogenetic perturbation in a monkey has produced a reliable report or movement on a trial-by-trial basis. And the reasons for that I don't think are well understood. I suspect it's something that we will master in the future. And this is certainly not a trial-by-trial basis. This is averaging over many trials. But I think this effect is on par with some of the others that have been seen, and it gives us hope that we'll be able to continue to use optogenetics and bring some of these genetic tools over to the monkey. Because in closing, I would just say that the experiments we've done in mice have really left me feeling like they are wonderful for certain classes of experiments. Detection, no problem. We can do that with natural stimuli or electrical activity that we induce with channel rhodopsin. We have also trained them to do things like target detection. They know vertical is the right target. They'll ignore everything else and respond when they see that target come on. But that's about where we run out. I would not try to train a mouse to do match to sample. I think it might be possible, but I'm just not sure it's worth the effort. And I feel like there's probably a whole range of cognitive function that goes on in primates, rhesus monkeys, and humans that are simply non-existent in the mouse. In wrapping up, I would just sort of run through some of the highlights here. Nobody's ever going to beat the mice for genetic tools. They're amazing. They work for amateurs. They're great. Monkeys have been difficult, but they are improving steadily. They will never be as good as the mice, but they will be, I think, increasingly useful and increasingly powerful in coming years. For simple perception, simple behavior, I think mice are just fine, and I'm actually very happy with the data that we've collected from our behaving mice. They're champs when they want to work, if you give them something that's within their range. But for the human condition, I feel like I just wouldn't bother. They're really just not going to give us the information we need. And actually, cost is not a factor in most of our decisions, but I thought I would just mention this. They're not cheap. My lab spends more on mice than on monkeys. By the time you have breeding lines and you're maintaining stocks and you have all these animals that are in training, it's a wash. And ultimately, when you're training animals for 2 months, the personnel costs are going to be a big factor either. So again, costs aren't the biggest issue, but they don't make the mouse a winner. So wrapping up, I'd just thank you, and I'd just like to acknowledge my colleague, Suprio especially, who did all of the monkey work that I've showed you today. Jackson Cohn and Mark Heisted did a lot of the work on the mouse side that I showed you here. And thank you.